Now I'm the IO state. So I'm very excited. Lab and everything. This work was done mainly by my graduate student, David Ream, and my programmer, Asma Kapoor, who's now at Iddo Friedberg's lab, actually. So the motivation for this work was basically to find out, and I'm glad that the organizers placed it like this, because the previous speaker talked about gene clusters, and I try to talk about how these gene clusters actually evolve. And you know, this is always a question: how do things do how do entities that do very complex things, like the eye or like any other organ or like gene clusters, evolve from the simpler building blocks? So I can immediately tell you that I really don't know the answer to that, but we discovered some very interesting things while trying to find that out. So I'll talk about operons. And operons are almost unique to prokaryotes. C. elegans has them too. And the idea is that you have several genes in the same strand that are co-regulated upstream by an operator and get co-transcribed in a single mRNA protein, also known as polycistronic mRNA, and then translated separately into the proteins. So as you can see, this is a very powerful mechanism to say for a metabolic pathway, for a protein complex. You get everybody translated together. This is great. Gene blocks are, you might say that operons would be a slightly more specific case of gene blocks. Gene blocks are any suspected syntenic group of open reading frames which have a maximum allowed spacing. And we worked with 500 nucleotide, but this is pretty robust. As you see from our work, we find this phenomenon also with anything between 0 through 500 through 700. Around 700 things start to fall apart because you can already throw in a little gene in the middle. But as long as you can't, those get transcribed separately and translated. But the idea is they can still be syntenic, meaning they can be. You can find them conserved in several other organisms. So operons are a very important feature in prokaryotic genetics. They often contain full metabolic pathways, a set of chemical processes that transform one compound to another. So studying operon evolution can help us understand many different things in microbial physiology. So we wanted to find out how these operons evolve, because we do see sometimes the genes as a gene block, sometimes as separate, and sometimes as a full-fledged operon. And we see them in very closely related organisms this way. And you know, when you think about it, we also see DNA and protein sequences in the same way. We actually, when we model the evolution of DNA or protein sequences, we have two events, evolutionary events, that we take into consideration. And we all know that. One is the mutation that you see on your left. Okay, on your left. And one is the indel, which you can see on your right. So these are the two events that we considered, and that's how we actually explain sequence evolution, you know, to everyone. Those are the two events that are used to explain sequence evolution. Now, we can do the same thing for gene blocks. We can look at other changes. There'll be more types of events, not just indels and mutations. And the events that we were looking at were deletions, meaning you have a gene that has been deleted from the block, a duplication, a gene has been duplicated within a block, and a split, meaning some of the block traveled elsewhere long enough on the strand to not be considered to be far enough on the strand or on another strand not to be considered to be in the same area of replication. Now, I'd like to say that there are, of course, other events that may happen, but we found that the other events that most other events you can imagine are actually correlated with these three events. So, for example, a split can also be, people would ask, okay, what about jumping on a strand? Jumping on a strand is always correlated with a split. You can say that a split can be on the same strand or on another strand, always correlated. So a split is a split. Doesn't matter if it goes on the same strand or the other strand. Other events are also correlated with these three events. So these are like the three basic events that we found that take place. So our aims in this work were first to determine gene blocks to determine the evolution of gene blocks. Sorry, that's a wrong misnomer here. And the other one is to use this determination of gene block evolution to actually find orthologous blocks and to reconstruct the ancestors. So how did we do that? Well, first we made a blastable database of all genes in assembled proteobacteria genomes. We started from proteobacteria because it's a simple, well-studied group. E. coli is there. E. coli is the best-studied genome in the world, except for some viruses. And we created a target DB from that. Then we used BLAST to find operon genes in target proteobacteria, okay? And operon genes are mostly, in most bacteria, the operon are computationally predicted. They're not experimentally annotated. The exceptions are E. coli and another bug, which are in Regulon DB, that we can see them. So, and then the idea is to compare blocks between target organisms by grouping genes spatially 500 nucleotide max. So the check mark, the green check mark shows you an actual two blocks, which we call orthologous blocks, and the X there shows, no, that is not a neighborhood. Okay. Graphically and manually, this is taken from a paper published in '96 by Fani et al. This is the histidine operon, the histidine biosynthesis operon. On top you see the E. coli. You can't read that. It doesn't really matter. As you can see, for all those up here, do I have a pointer here? No, doesn't. Oh yeah, there's something. Yeah. So for all those up here, these are all the gamma proteobacteria. There's a series conservation, mostly of the operon as well. Every time you see a tick up here, that's an operon. Okay. And, but down here you see still some sort of conservation, especially in this family, but here you see all sorts of split apart of the component genes. They're no longer in an operon, they're definitely not even longer in a block, they're sort of spread all around. But there is an evolutionary trend here, and we'd like to sort of quantify that and find out about it. So a new term you should all use, the ortho block, and that merges two terms, and we combine informaticians love to make up terms, right? So orthology, which is homology due to speciation, the structural genome, and these are two descendants. This is speciation, and we have two different blocks here, and a gene block, which was defined earlier. So an ortho block is a gene block where each gene is orthologous to a reference gene block. This gene block is located in a target organism. This is a reference organism. And these would be considered to be orthobloxus genes, or whatever you want to call them, genes on an orthoblock. Even though some of the genes disappeared here, were deleted along the way, these genes are still blocked together, and they're considered to be the orthologs, the true orthologs. Whereas here, these might be orthologous, but the blockishness has gone away, so we don't consider it to be one. So how do we select the true orthoblock? Because selecting a true orthoblock means finding the correct orthologous genes to the operons that we are studying. So up to this point, BLAST recovers a large number of homologs for each operon, and we all know this problem. BLAST is incredibly precise, but can also be incredibly noisy. And we organize each hit by the target organism. Most of these hits are spurious, and we need somehow to filter them out. We do that using a greedy algorithm. We take an operon, like I said, that was experimentally determined in E. coli using Regulon DB. And to select the true orthoblock, we blast it against another organism, something else, S else, okay? We blast it against S else. And homologs that we find to this operon A are all of these. Now, how do we tease out the correct orthoblock from that? So we looked at the number of events that are needed to reach each orthoblock candidate. That means, these are all the orthoblock candidates that we have here. That means that we reduce the number of splits, duplications, and deletions, those events, right? The whole idea of evolution is that, these event-based study of evolution, is that you're supposed to reach some sort of minimum, okay? So we looked first at this possible orthoblock, but here we have 2 splits, 0 deletions, 0 duplications, total events of 2. We looked at this candidate for orthoblock. We have 1 split, 0 deletion, 1 duplication, again, total event of 2, so nothing is really better here. We look at this candidate for the orthoblock. We found 1 split, 0 deletion, 0 duplications, total event 1, and this would be our winning candidate for the genes that are orthologous to the genes that were in the reference. Okay, so that is what we found. Right, fantastic. So this is how it looks genomically, of course. And in the final result, we wrote this software where you can insert a series of a bunch of basically GenBank files, okay, reference GenBank files, and a reference operon from E. coli. E. coli is down here. You can't read this probably, but E. coli is down here in the bottom. This is Shigella, Salmonella. So here we have all the gamma proteobacteria. Here are beta proteobacteria, epsilon proteobacteria, and alpha proteobacteria. These are not all of them. These are representative from these families. Okay? And here you have the operon in E. coli. You can see that it's well conserved. This is the operon Shigella, well conserved. And this is the same His operon that was done manually by Fani et al. in '96. We did it now. We built this picture now automatically. So we kind of reconstructed their research. It seemed pretty cool. So we were happy. And you can see these various splits here and strandedness and and so on and so forth. We call these RV diagrams Asma-Riaz visualizations. Asma-Riaz was the programmer who did this. The other thing we can do now with each and every operon in E. coli is find out, how conserved it is in our corpus of gamma proteobacteria that we are looking at. So here we have highly conserved operons, and I'll talk about them in a minute. And these basically have very few events. This one has just a couple of deletions, a few splits, and no duplications. This one has very low frequency. This is a normalized count, of course, so we can compare everybody. Low number of deletions, very small numbers, and maybe one duplication normalized. Whereas here we have highly non-conserved operons. These are sort of spread all over the place, usually not operons, usually not conserved at all. And, oops, sorry. What did I do? Okay, yeah, all right. So, those are very non-conserved. So we have actually a quantitative way of looking at which operons, which gene blocks are conserved as blocks within the corpus that of organisms that we're looking at versus which ones are not. And here we have in the blue, those are the more conserved. This is counterintuitive to American politics, for those of you who know the color blue and red stand for. The more conserved are here. This is actually RPOB, which everybody knows. This is RNA polymerase B, an incredibly conserved RNA polymerase complex. Everyone knows that's conserved. Another one that we found that's highly conserved is, of course, this is the ATPase, and we'll talk about these two later. So this is the ATP, and this is the ATPase complex. ATPase is a highly conserved protein complex throughout life, and it was not surprising that we found that it was really conserved here. And here we have a heat map that shows the number of deletions, duplications, and splits. So no duplications, very few splits, very few deletions. The main changes are between the alpha epsilon group and everybody else. So that's kind of interesting to see how these things evolve. And these diagrams give you a really nice idea of how many events and the frequency of events that you're getting between each and every block that you have. So this is for the ATP, fairly conserved. This is what a fairly conserved orthoblock looks like. And also RPOB, again, incredibly conserved. You don't see many events running around here, no duplications, only one deletion, which was actually a false deletion. We just didn't find the actual ortholog because our blast threshold was pretty high, 10 to the minus 10, and these are the splits. Okay, great, fantastic. Here, so we've seen what two conserved orthoblocks look like. What do non-conserved orthoblocks look like? Well, they look like a mess. After seeing this, my wife said, "Oh, that's like in Burda, that knitting magazine." So these are like, so she was really happy. She called them Burdagrams. Hydrogenase 4 complex, and this complex apparently is an operon in E. coli. Okay, we have an operator, we have all the genomes. It's hardly ever expressed. It's not expressed under any known growth conditions. Nobody actually managed to express this. The only way they managed to express this was in the lab when they actually stuck a promoter in front of it, some sort of phage promoter that got it to run. So this is, but it is an actual operon, even though we don't know exactly what it is. But in E. coli, it is an operon, and everybody else, it's not really. And that is how you see that it's totally not conserved here. And you see events popping up all over the place, including the green lines. And the green lines mean simply that that the genes are simply not there. We couldn't find any orthologs in those species. This is also a non-conserved one, where mostly deleted. Anaerobic L-threonine degradation to propionate. Not a lot of bugs have this particular metabolism. E. coli has it. A few gamma proteobacteria have it, and that's about it. So we want to see whether there is some sort of correlation between the function of these operons, the function of what the operons do, and their evolutionary conservation. That's a little bit subjective, right? Assigning a function to a gene is pretty hard. Those of you who know me know that this is what I've been doing for 10 years, and trying to do that automatically, and that was very tough. And assigning a function to an operon is even worse. But we tried. We said, okay, let's take 6 categories: information, molecular complex, metabolism, stress response, energy, and environmental response, and try to assign those as best as we can. And what we find is that the more conserved operons have to do with either molecular complex, these Legos here, or with information processing, such as RPOB. And the less conserved operons have to do something more with stress response, environmental response, kind of like what we expected. Trying to do a non-parametric statistical correlation analysis, we didn't come up significant, so this is just an overview. This is a hint of things to come. We're not exactly sure that this is, but it's nice. It's a nice first preliminary finding. So the conclusions are that we observed local conservation of events, that when educated by phylogenetic tree makes sense. More conserved gene blocks tend to include core metabolism. Less conserved gene blocks tend to include niche species specialization, such as accessory metabolite catabolism and things that are specific to bugs in specific conditions. Okay, so the other thing that we wanted to do is, can we reconstruct ancestral gene blocks, not taking into account horizontal gene transfer, which is rampant in bacteria, so keep that in mind. We want to see whether through the normal vertical gene transfer, whether we could construct ancestral gene blocks. So given to observed gene blocks, what is the most likely series of events that have occurred since their common ancestor, by minimizing the events. So we use the maximum likelihood to optimize for the lowest cost. So let's say this is our ancestral gene block. We have a cost and deletion here of -1, -5. So this would not be the gene block, and this would be the ancestral gene block simply by going to organism 1 and organism 2 that are extant, down here. All right. So we can actually reconstruct the tree using this algorithm from 1 and 2. 1 and 2, we find this is the best candidate. 4 and 5, we find this, and so on. Trouble is, when we actually step out of toy data and start working with real data, the tree starts blowing up in terms of possible candidates. We are trying to reduce that space now. So again, this is work in progress. So summary of the ancestral reconstruction: we can report the structure of an operon found in a target organism. We can overlay a tree. The cost function helps us to estimate the ancestor as a better choice. And algorithms have been developed to make this computation feasible. We're still reducing search space. It's still problematic. So these are the general conclusion. We can computationally determine gene blocks in extant organisms. And when doing the events that we identify can be used to compare one gene block to each other. We can also use the events to compare organisms within gene blocks. We got some really cool results when doing this. And results support the idea that this work can be used to construct ancestry of gene blocks to determine evolutionary directions. And this work was published in Bioinformatics. I'd like to acknowledge David Ream, the grad student who did most of the work and will be graduating very soon and looking for a job in case anybody's interested. Asma Kapoor, my programmer, who is now with Aviv Regev's lab, and Jake Gray, who did quite a bit of the coding. NSF provided the funding. Like I said, just a little plug here, I'm recruiting into my lab. I'm at Iowa State University now. If you're interested in this kind of stuff, we're doing it. If you're interested in protein gene block evolution, function prediction, other things, I'm looking for postdocs, programmers, grad students. Ames is a great place to live in, and Iowa State has a really, really strong scientific and technical environment. It's a joy to be there and play with all their toys. Talk to me. You can find me using these. All right, thanks very much.
What happened to the delta or proteobacteria in your experiments? There's experimentally determined operons in D. vulgaris, which might be...
Yeah, very few of them. We just went with one gold standard to sort of standardize the standard, as it were. Now so when we developed it, we decided let's do it simple and just use one gold standard. Now we have the software to actually take any other organism, whether it is experimentally determined or not, and use that as a gold standard.
So you use E. coli and B. subtilis as your base?
We used E. coli because it was the easiest thing to do at the time. Yes? Eukaryotes. Eukaryotes mostly don't have operons except for C. elegans. They have blocks. There is some work done on that, not ours. Pavel Pevsner and one of his students, whose name I forget, who's now a professor at DC, and I can show you some of that work if you're interested in how this is done in eukaryotes. So it's a similar but quite different ballgame. Okay. Any other questions? Oh, sure. Shall I step down? Shall I go down?
Thank you very much.